Good afternoon. My name is Dr. Nicole Nietlisbach, and I'm a veterinarian with the Wisconsin DNR focusing on fish health. Today, I'm going to be talking about smallmouth bass, specifically focusing on some health issues we've seen in bass in the waters around our county. Since about 2008, we've received reports of skin ulcers in adult smallmouth bass. Most of these reports come in from anglers, especially anglers involved with tournaments, sometimes guides, and they usually come in during spring. We sometimes get reports in late summer, early fall. There hasn't been any associated mortality with these disease events, and it's been noted that more often the larger adult smallmouth bass are the ones that have the skin lesions, and not as many of the smaller fish. This has especially been noted by biologists during surveys. And this is just an image of a fairly typical lesion. They're usually circular like this and very shallow, and it can appear anywhere on the body, but are often kind of on the sides of the body like this. So similar issues in the same area have been investigated over the years multiple times, and looking back at historical case notes, there's been very little consistency in what pathogens have been isolated from year to year, and even between fish during the same outbreaks. There's been a plethora of bacterial pathogens that have been isolated, some of which do cause skin disease. So Flavobacterium columnare, which causes columnaris, and different little Aeromonas species, which also can cause skin disease. There's also been a few different viral isolations of note. VHS was isolated once early on and hasn't been repeated again. And then there was some evidence of an orthomyxa-like virus via next generation sequencing, which is an advanced genetic analysis technique. I'm not very familiar with how that works, so I won't try to explain it. That has not been repeated again either. There's also been a little histology done, so that's looking at the tissues on a cellular level with stained slides. And the common finding over the years has been something called granulomatous inflammation, which is often a type of chronic inflammation. And so this past fall, we got another opportunity to sample fish. This was my first opportunity to sample these fish. I've been with the DNR for only about 3 years. So smallmouth bass with skin lesions were reported by anglers, a lot of guides, in late August and early September. And we were able to get samples collected from some of the fish turned in by guides. Scott Hansen, the biologist at Sturgeon Bay, was able to collect some samples and send them to the diagnostic lab in Florida that was able to do some PCR work to look for a few specific viruses. Myself and my colleague, Dr. Goddard, also a fish veterinarian for the DNR, were able to sample some fresh, still living fish that Scott was able to acquire for us, and we were able to do some more extensive diagnostics on those fish. And these fish came from a tournament that had an estimated 30 to 35% of fish at weigh-in showing skin lesions of varying severity. We were able to look for parasites. We were able to collect tissues for trying to isolate viruses on cell culture. And we were able to collect samples for bacteriology and also saved a lot of different tissues to look at via histology. So just to give you an idea of what we saw that day, this is a fish with a pretty typical lesion. Again, very round. Most of the lesions that day had a pretty pale center of necrotic tissue and were often lined kind of a red rim. This is a closer look at a typical lesion. And again, you can see that pale necrotic tissue in the center and then red, very inflamed outer edge. Some of the fish that had these skin lesions would also have involvement of their fins. And so you can see in this particular fish, there was also some erosion, kind of melting away of the tissue of the pectoral fin that was overlying the main skin lesion. This is an example of a fish with slightly obvious lesions. You can see right here, there's a little bit of scale loss, and here as well, and then a little bit of redness. Internally, we saw some possible changes. The spleens, at least to me, looked a little bit big, though I don't get the opportunity to sample adult smallmouth bass that often. So it's possible that it's a little bit more normal for them. A little bit hard to say. Interestingly, two of the 10 fish that we sampled had these weird kidney masses. I don't know if you can see that here. We don't really know whether that was just kind of a random finding or if it had anything to do with the disease. It's a closer picture of that kidney mass. And some of the fish had some changes to their swim bladders. And so in this one, the swim bladder's open and we're looking at the lining. And you can see some bleeding around the vessels in the lining. And it's possible that you could get similar lesions from fish that are brought up from depth. So that might be explanation for what's going on here, or could have to do with whatever disease process was going on in the fish at the time. There's also a pretty good view of a lot of the fish have very pale livers. They're kind of yellow, and that can happen with fish. Hard to say whether it was related to both the skin disease or not. At least two of the fish had some eye abnormalities, and so this fish, you can see how cloudy the eye looks. This fish had that on both sides, and that can be a sign of inflammation in the eye. And one fish had what we call coelomic adhesions. So you can kind of see this spider web type material kind of attaching the fat in the abdomen to the lining of the coelom, and this can be a sign of general inflammation within the body cavity. So the most interesting finding from this past year was that we found a virus called largemouth bass virus in all 14 of the 14 fish sampled. We found it in skin samples via PCR, and we also found it in organ samples via PCR. And we were able to send internal organ samples, which included kidney, spleen, and swim bladder, to the U.S. Fish and Wildlife Fish Health Lab in La Crosse, and they were able to isolate largemouth bass virus from those tissues on cell culture from all 10 of those fish that we submitted. So we also, just to talk about the rest of the pathogens that we found, we again, consistent with past years, found a number of different bacterial species present, some Aeromonas species, some Flavobacterium species, and others, and they weren't consistently on samples from every single fish. So just a few fish would have Aeromonas, a few would have Flavobacterium. It was pretty inconsistent. This picture up in the right-hand corner is of our bacterial plates and kind of what the colonies end up looking like. So what is largemouth bass virus, or LMBV? LMBV is a member of the Iridoviridae virus family and is currently classified in the ranavirus genus. Iridoviruses are generally large viruses that are known to infect ectothermic animals, sometimes insects, not known to be a threat to humans or other mammals. LMBV, as the name suggests, is most known for being able to infect and cause disease in largemouth bass. And the signs are pretty vague. They can involve lethargy, trouble swimming, and floating at the surface of the water. And on internal exams, the fish will sometimes have overinflated swim bladders, like this picture, and sometimes swim bladders with a yellowish fluid inside them. The virus has been isolated from a number of different species, including other shrimp-trachid species like smallmouth bass and bluegill. And anecdotally is more often isolated from fish without any signs of disease, even largemouth bass. And just a little history about the virus. It was first detected in Florida in the 1990s. and the first actual fish kill associated with the virus was in South Carolina around the same time. There is some thought that uh, its source is from the ornamental fish trade. There's two ornamental fish viruses that are really closely related to largemouth bass virus, one from guppies and one from doctor fish. And so that's sort of a theory about maybe how it got introduced to the United States. But since the 1990s, it's spread all over the eastern, um, and southern, and central United States, and is in at least 22 states at this point in time. The detection of LMBV has really brought up more questions than answers for us. There's very little literature on the way LMBV behaves in smallmouth bass. There is one great paper out of Michigan that looks at exposing young of the year smallmouth bass in a lab setting to different isolates of LMBV, and they did see that the virus was able to cause disease, and in some cases was even able to cause skin ulcers, which looked pretty similar to what we had seen. But with just one study, and considering that the fish were young of the year and that the temperatures were different than what we were seeing in water temperatures around Green Bay, Door County, during our outbreak, it's hard to make kind of conclusions and extrapolations from that study. But certainly is evidence that supports the thought that LMBV was probably a main player in the disease outbreaks this year. We do have questions about, what is the relationship between LMBV and the bacteria, different bacterial pathogens that we isolated, and environmental and angling stress? Was the virus there first and causing disease and immunosuppressing the fish, allowing for bacteria to come in and cause skin lesions? Did the bacteria show up first and start causing disease and allow the fish to be infected with LMBV? Did something about the angling stress or the water temperature allow the virus to activate and cause disease? There's just a lot of different scenarios and ways that these different factors can be playing to cause the disease outbreaks. And so we can't really say for certain how they're all interacting together. We also question why we haven't detected largemouth bass virus in the past. We've seen really similar disease signs in past outbreaks. Did we just not collect the samples correctly? This year we decided to include swim bladder with some of our fresh tissue samples. Was that a better tissue sample for detecting the virus on viral isolation? Or was this just a kind of a one-time thing? Is there a different pathogen or different pathogens and environmental stressors that are causing similar looking lesions every year? There's a lot of fish pathogens that do cause similar looking disease signs, and it can be a really frustrating part of diagnosing primary causes of disease in fish. And we wonder, is this largemouth bass virus a new problem? Has it been here? And we also wonder whether the virus has caused disease issues in other parts of the state. As I mentioned earlier, it's thought that LMBV is ubiquitous, and if it is, are largemouth bass in other parts of the state also developing skin disease? So all these questions that we're hoping to answer going forward, or hoping to at least work towards some better understanding, even if an answer is a real answer is hard to come by. After the detection this fall, our focus has been and will continue to be communication with the public. This is a high-profile bass fishery, and there has been a lot of public interest in these disease outbreaks over the years. So we've released a press release this fall that gave basic information about the detection and also tried to give some guidance to anglers as far as best practices. We also are planning to add an LMBV page to the DNR webpage. Dr. Goddard and I are working on that. We'll hopefully have something soon. In moving forward, as I outlined previously, we have a lot of questions that we still need to answer. We definitely want to focus on seeing if we isolate largemouth bass virus again from similar skin disease outbreaks in smallmouth bass, especially in the area around our county again. We also just want to get a better idea of where largemouth bass virus is and if it's involved with any similar disease outbreaks in smallmouth bass in other areas of the state. We have detected largemouth bass virus in the past, or previous colleagues have. Lake Geneva and Eagle Lake have had largemouth bass virus show up in largemouth bass that weren't experiencing disease at the times. U.S. Fish and Wildlife Service has done quite a lot of work, survey work for largemouth bass virus, and over the past 2 decades, they have detected it in the Mississippi River in multiple pools along the Wisconsin border, and also from other inland lakes around Wisconsin. So we know the virus is here, but we'd like to get a better picture of where it is now and if it is causing other disease issues that we are unaware of. We in talks with U.S. Fish and Wildlife Service and other colleagues, it seems like it's likely fairly ubiquitous around Wisconsin. But again, we would just like more information and see where it actually is. So as we hopefully answer questions in the future that we've had, we will have to reevaluate what management options we have and what management decisions we can make. It's really difficult to control pathogens in the wild. And so realistically, we might not have many great options um, in front of us. But for now, we have tried to, in our press release, put out different recommendations and best practices for tournaments and anglers. And those recommendations really focus on trying not to spread pathogens, especially largemouth bass virus, to other water bodies that may not have it yet. Even though we think it's possibly ubiquitous in the major water bodies in Wisconsin, it's possible that there's inland lakes that don't yet have it and could have naive populations of largemouth bass or smallmouth bass. So, being sure not to transport live fish or to transport the virus with equipment that hasn't been disinfected yet. A lot of the same disinfection recommendations that are out there for VHS will work for largemouth bass virus, and so we're sure to include links to those guidances in our press release, and we'll do so going forward. We've also tried to include recommendations that hopefully make it so that it's more likely that the fish that are caught are going to survive. When the water is really warm, not only do you have lower DO issues, you also might see the virus have more pathogenicity. Looking at a lot of the literature on largemouth bass virus in largemouth bass, and also the study out of Michigan in smallmouth bass, largemouth bass virus seems to be more pathogenic in warmer water. So if tournaments can avoid fishing when the water is warmest, that might be helpful in limiting the impacts of largemouth bass virus or other pathogens. Besides focusing on communicating best practice techniques and everything and trying to reevaluate our management decisions, it's really important that we focus on collaboration with our colleagues. Myself and Dr. Goddard are on the Great Lakes Fish Health Committee, which has representatives from state, federal, and tribal agencies from around the Great Lakes Basin, and we're actively having communications about this. Some other regions have experienced similar things with smallmouth bass, and so we're actively communicating about this, and that's really important for figuring out the best next steps. There's a lot of great research and fish health minds in that committee, and I think a lot of good ideas for going forward will come from that. And here are just some resources that I took some information from. just want to quickly acknowledge that a lot of people have done a lot of work and have provided a lot of guidance and information on this issue. So big shout out to all of them. Thank you very much, and I look forward to your questions.